Hey everyone, Dr. Jack Audie here, and before I jump into bacteria and pathogens, what are they, how do they cause disease, I want to jump into a technique known as gram staining because it's so fundamental to bacterial biology and how we talk about bacteria that it's kind of best to start with gram staining than it is to jump into bacteria as pathogens. So let's jump into it. So here we have a scanning electron micrograph. An electron microscope is just a more powerful microscope. It uses a different technology to light microscopy, so it allows us to see even smaller structures like bacteria. The first thing that we can see is that there's different shapes, right? Which is a fantastic way to break up bacteria into different groups. So as we want to explore biology and how to deal with these pathogens and how do they cause pathogens and what's in common between bacteria, we need to break them up into little groups, species if you will. And one way we can do that is a size and shape, right? So that there's different sizes and shapes. But there is another way, so we can break them up here based on shape, but there is another way to break them up called gram staining. And what's fantastic about this is it actually tells us, gram staining tells us something about the biology of that bacteria, as you'll see as we jump into how gram staining works. So it groups the bacteria instead of by shape, by whether or not they stain for this purple stain that's called gram staining. So this purple stain down here, let me actually bring up the laser pointer, this stain down here, this is positively gram stain, so this is gram positive bacteria, and this pink is actually a counter stain, it's not the gram stain, and that is a gram negative bacteria, this pink stain here. Yeah, so gram positive, the purple, gram negative, and it's an important way to break a bacteria. It tells us whether an antibiotic is going to be more effective. It tells us when they were related in evolutionary history. It tells us so much because it's fundamental to the biology of the bacteria. So first I'll do a run through on what happens during a gram staining, then I'll explain how that works, and then we'll look into the biology of it, right? So step one is we put on a crystal violet stain. This is the purple gram stain. Now the important thing is it actually stains all the bacteria. So all the bacteria initially stain positive for the gram stain, the purple gram stain. We then put in iodine. Now iodine reacts with the crystal violet to lock it in place, okay? So it almost fixes the crystal violet in place in the stain. Then we run an alcohol or acetone over it. Now alcohol or acetone will start washing out the gram stain, and it will wash it out first from the gram negative bacteria. So the gram negative bacteria will now have the gram stain completely washed out of them, and they will become clear, while the gram positive staining, if we do the washes at the right time interval, you don't wash too long, the gram positive bacteria will retain the stain and will retain purple. We then do a counter stain. There's a number of different kinds, but safranin is one of those. So we end up with, and that's a pink stain, and that will actually stain both groups of bacteria, but because purple is darker and more visible, we end up with a purple gram positive bacteria and a pink gram negative bacteria. So let's look at the biology. How does that work? Well, to understand the biology, we need to understand the cell walls and the major kinds of differences in cell wall structures in bacteria. So here we have a gram negative bacteria and we have a gram positive bacteria, and we've zoomed in to look at the cell walls of these two bacteria, and we can see that they're actually a bit different, quite a bit different going on here. So let's break it down. The first layer is the phospholipid bilayer, which we've all got. We've got phospholipid bilayers, plants, fungus, bacteria, all have this phospholipid bilayer. And it's just that hydrophobic, hydrophilic interaction that creates that phospholipid bilayer. And this is a flexible layer that controls what goes in and out of the cells. And that's why we've all got it. It's a very important biological structure. Now, this is unique to bacteria. So we have, this is a defining feature of bacteria, I should say. And this is the peptidoglycan layer. Now let's break down those words. Peptido comes from peptide, and peptides are just small proteins, and proteins are just made out of amino acids. So a peptide is just a chain of a few amino acids. Glycan means a chain of sugars. Think gly and glue. So there is glucan, which is a chain of glucoses. Glycan means a chain of a mix of sugars. So there's more than one kind of sugar in there. Now these sugars, I should say, are hexose. So glucose could be in a chain or it could be in a little hexagon, right? And most sugars have that option of being in a chain or in a little hexagon. So a hexose is that hexagonal sugar. And you can see the cell walls in the diagram made up of chains of hexagons all joined together. So this chain of sugars is chains of hexose, hexagonal sugars linked all together. And then they're sort of cross-linked. So the chains of sugars are sort of cross-linked with the little peptides. It's sort of tied all together. You don't want just a bunch of ropes. You want those ropes connected together in a mesh-like structure. Now these are rigid, so peptidoglycans are quite rigid, and they need to be rigid, right? Because this is how bacteria create different shapes. So there are spiral bacteria, there are rod bacteria. You could not create those shapes with a phospholipid bilayer because a phospholipid bilayer is flexible, right? So these rigid outer walls allow bacteria to make these unique shapes which help them survive in certain environments. The other thing they do is make bacteria very robust. If you were to take my cheek cells and smear them on the desk, with a swab, smear it on the desk, those cheek cells would die very quickly. Whereas bacteria are more robust in the environment because they have this rigid cell wall, which gives them resistance to stresses like osmotic stresses, which are a very important stress that will cause popping of cells when they're in an environment they don't like. Yeah, so if you put my cheek cells in fresh water, they will osmotically explode. Any of my cells in fresh water, they'll osmotically explode, whereas bacteria are resistant to that because they have that rigid cell wall that protects that expansion and that popping that can happen under osmotic stress, like being in a freshwater solution with no salts. Okay, yep. So then the gram-negative bacteria then have a second phospholipid bilayer, which actually creates some interesting biology and kind of lines up with things like mitochondria, those two phospholipid bilayers. So there's some interesting biology around that. But that's the basic structures of the two types of bacterial cell walls. Now let's see what happens to those walls as we run them through a gram staining. So we start with the crystal violet. The crystal violet is positively charged, which makes it want to interact with the negatively charged peptidoglycan layer. So it comes in here and it interacts with those layers. But right now, the crystal violet is still water soluble. So if we put that in a water solution, it would wash out. So yes, now the cell walls on both sides have stained purple. And now we put in iodine. Now iodine's negatively charged. It interacts with the crystal violet, creating a crystal violet iodine structure. This makes it not soluble in water, but still soluble in alcohol and acetone, okay? So now it's resistant to being washed out, okay? So it kind of fixes that crystal violet, the purple crystal violet, in place. Now we do an alcohol wash or an acetone wash. Now alcohol and acetone make membranes permeable, so the outer membrane of the gram-negative bacteria becomes permeable. It becomes not an issue. And now the alcohol and acetone will start to wash out the gram staining, the crystal violet iodine complexes. And it will start to wash it out of both, but because of the thickness of the peptidoglycan layer, the gram-positive bacteria take longer to completely wash out the gram staining. Now you could keep washing in alcohol. Actually, it doesn't take that long to wash out the gram staining in both bacterial types. So it has to be a very brief wash with the alcohol or acetone. And if you do that brief wash, it washes out first from the gram-negative bacteria before the gram-positive bacteria, because the gram-negative bacteria have such a thin peptidoglycan layer. Now we do a safranin counterstain or a number of other counterstains, and this actually stains membranes, and so it stains both the gram-negative and gram-positive bacteria, but because purple is a much darker, stronger stain, we don't really see it in the gram-positive bacteria. So now we end up with pink, gram-negative, purple, gram-positive bacteria. So you can see what we're doing here. We add crystal violet, that stains them purple, add iodine to fix it in place, add alcohol to wash it out, but it washes out first from gram-negative bacteria, and then we add the safranin counterstain, and that's where we end up. Right, so up next, in the next video, I'm going to take you through the structure and biology of bacteria before we jump into some examples of bacteria as pathogens. Thanks, guys.